on transport chain, right? So these guys are not using oxygen, right? So is that anaerobic or aerobic respiration? Anaerobic. I heard it in the yawn. All right, so anaerobic respiration, dedication, right? So yeah, make sure you are familiar with those processes, right? Just keep ourselves like updated on our processes and how we see that in our bacteria. All right, so this shows us a picture on this slide, figure 11-10 of the bacterium, the Vibrio attacking this gram-negative bacterium right here. So like I was mentioning to you guys, it's going to penetrate that cell's wall, outermost wall, and try to get into the periplasmal space to ultimately divide. It's going to burrow in there, hang out. What's funny is that when it does burrow in there, it's going to lose its flagellum too. But once it loses its flagellum inside of that periplasmal space, it's going to start making copies of itself, getting a little bit bigger, and ultimately taking over that cell. It's going to kill that cell. It's going to take it out. All right, so myxococcytes. So this one, myxo, is going to tell us something about mucus. All right, so it tells us something about mucus, and the ALES suffix then tells us that it's what? It's in order. All right, good deal. So this is a soil microbe too. So we found this one in the soil. And one thing that's really cool about this one, before we jump into like how it actually looks, is that some of these bacteria can actually produce this anti-cancer agent called epithylones. So epithylones are anti-cancer agents, and the really cool thing about it is that these guys can target and help obliterate those breast cancer cells. And this is a naturally producing chemical, right? So it's actually involved in cancer chemotherapy, right? So when we talk about chemotherapy, a lot of times you automatically think like it's going to be lab synthesized products, but it's really not. Some of those chemical cancer chemotherapy treatments are going to be things that are naturally found in the environment. So taxanes are naturally found in the environment. That's actually, taxanes are a group of cancer chemotherapy drugs. They're naturally produced. We discovered those in the 1960s. Well, not we. Scientists discovered those in the 1960s. I wasn't there in the 1960s. My mama was there. Well, she wasn't even there in the 60s. Well, she did. She got there, but like not in 1962. But yeah. So we're talking about this though. It's pretty cool that these chemicals are produced by this particular bacterium, the soil microbe. And the thing that makes it so cool too, though, is that it's made by an organism that for this particular chemical. And so that tells us a lot about the microbes that we have. So a lot of our microbes, or not microbes, a lot of our antimicrobials come from the soil, right? We know streptomycin, on our last exam, streptomycin is responsible for producing 2/3 of the antibiotics that we actually have today. And so just knowing this information about like how soil microbes are so important is why we have a lot of research right now going on with soil microbes. Like there's something I want to do probably in the future with my students here, with using soil, just to see if we find anything like really cool or funky going on in the soil out here in California, right? Because soil is going to be very beneficial with treating those different infections. So yeah, this particular microbe involved with producing the chemical epithylones that has anti-cancer activity. And it's really good for treating breast cancer. So these cells also can aggregate or cluster together to form a fruiting body, which is really interesting because fruiting bodies are normally seen in fungi. So fruiting bodies are normally seen in fungi. So it forms a fruiting body and it contains myxospores, and spores are normally seen in fungi as well, right? So this is a really interesting process for this particular bacteria. So let's look at what this looks like on the next slide. All right, so figure 11-11 is going to show us the life cycle of this particular bacterium. So it shows us the life cycle of this particular bacterium. And so this is very similar to what we see in slime molds and fungi. So this kind of looks like a slime mold, to be honest. So when looking at this, and we'll learn a lot more about slime molds in chapter 12, 13, in the eukaryotic chapter. So what we have here is a scanning electron micrograph of the mycobacterium fruiting body. And on the very tip of it, it shows these little spores, the myxospores, on the edge, those small circular structures. So these myxospores are going to be present. And so looking at this particular, they're going to be released from the fruiting bodies when the conditions are favorable. So once they're released, they'll be able to land on different surfaces. They'll be able to grow, germinate. They're going to form the gram-negative vegetative cells. All right, so vegetative cells are those that are like the resting cells. They don't really have any, well, if the cells had endospores, you have your vegetative cell with the endospores on the inside. These guys, not so much. These regular old vegetative cells that are ready to divide, make more and more copies of themselves. These guys will ultimately grow, and as they grow, they begin to cluster together, they swarm together to make a more solid structure for your fruiting body, your early fruiting body structures. They continuously do this So when you have your fruiting body, that's gonna be the mature structure. That's gonna contain all those mixed spores in these different sacs called the sporangioles. And so within the sporangioles, you have all the spores that'll then just like continuously be released and make more and more and more copies of themselves as the conditions are favorable. If the conditions are unfavorable, they won't be releasing those spores. They will not be going through the cycle, right? So in favorable conditions, they will make more and more and more and more copies of
Is that
themselves.
kind of similar to fungi?
Yeah, it is. That's what I was saying. So like these guys are very similar to fungi. They're not fungi.
Bacteria.
Yeah, they're bacteria, but so that's one thing that's really interesting about how our different cells are sometimes more similar than they are different, right? So like we've seen some instances where we have like, for instance, Mycobacterium has this like it has the waxy lipid structure with it, which is kind of similar to behavior with fungi. So it's like we have certain similarities that exist, right? But they're not fungi though. This behavior is like them. All right. So what do I want you guys to know for the Delaporteobacteria? So I would like you guys to know for this particular one, the myxocapalities, that one's gonna be really interesting for understanding our medicinal value for that particular anti-cancer agent. And then for the fruiting body, the process, the life cycle. The life cycle is what you really, really want to focus on. And so the sulfovibrinales, that's cool. That's the fact that they're sulfur-reducing bacteria is cool. The Vibrio, I would remember that one only because it is a predatory bacterium. Typically these Delta proteobacteria are predatory. So this particular class of proteobacteria, they are predatory. And so the Delta Vibrio though is really interesting in that it can target those other gram-negatives, because it's gram-negative in itself. That's what makes it really interesting. So it's able to target the gram-negative bacteria, just like it's a gram-negative, and destroy those gram-negative cells. All right. So you guys want to make a key, knock yourselves out. I'm not making a key of those three, but you can. All right. So then we have the last group for the proteobacteria. So I put it in very small writing. Formerly the epsilon proteobacteria. It's now the Campylobacteriota. All right. So this particular class of bacteria, these are still gram-negative bacteria. They're slender in their appearance. They're rod-shaped, and they can be either helical or curved. Okay, they can be either helical or curved. And the thing that's really cool about this is that you guys see these every week in the lab on my PowerPoint presentation. I always have a picture of Helicobacter as like the little green border outline. The bacteria, that's what that is. All right. Let's talk about this. Microaerophilic. What does that mean? Microaerophilic. Never heard of that. It's a little bit of, not air, but a little bit of oxygen. Great job. Yep. All right. So the reason why I always say not air, because within the air we got a lot of stuff that's in the air, right? Like we got some little nitrogen, we have oxygen, we have carbon dioxide, we have a lot of stuff in the air. So yeah, a little bit, small amounts of oxygen. So that's cool. All right, so then we have Campylobacter. So Campylobacter is microaerophilic. It's a Vibrio shape, so it's that comma-shaped one, right? So it's like a little comma-shaped microbe, and it has one polar flagellum. So just one polar flagellum, right? And the model example is Campylobacter jejuni. So Campylobacter jejuni is called the foodborne illnesses for individuals that they're affected with that one. Now, we're talking about Helicobacter. Helicobacter is pretty cool. Helicobacter was actually just discovered. Helicobacter pylori was discovered in 1982. Yeah, because I wasn't born yet. Close. So in 1982, they discovered Helicobacter pylori. There's a really interesting story behind it. Two gentlemen were working on it in the lab. I forgot their names. I know one's name was Barry. And people weren't really taking their research seriously. And this man decided to drink a broth. So he said that he was able to treat, crazy. So he treated somebody with the bacterial infection. He treated somebody with the infection with an antibiotic, but he wasn't able to show the effectiveness of the work, which was really weird because it's the 80s and the 90s. So I'm like, okay. So he ended up the next day took a broth of that sample. He got the sample from the patient, the Helicobacter pylori sample from the patient, grew it in some broth medium, and he drunk it. Don't ever do that. Do not trust your work that much. Don't do that. And so he did it. He drank it. He got really, really sick, very ill. And he ended up treating himself, and that's how the work kind of got discovered. So he got a Nobel Prize in physiology for the work. Him and his collaborators, to 2005 or something. Yeah, crazy stuff. But yeah, so that's how they discovered Helicobacter pylori, which is the causative agent of stomach ulcers, right? The peptic ulcers that you can potentially have. You don't want those, but that's the thing. That can unfortunately cause stomach cancer as well if you don't treat that, right? Now, interestingly enough, if you look at the pylori, some people carry it in small concentrations in their gut naturally, right? So some people can actually have it present in their guts. And the thing with that is, would be if the good bacteria are overcame by the bad bacteria, that's why you see the proliferation of those guys, and that's how you see the infection. So these are microaerophilic curved rods, and they have a lot of flagella, multiple flagella present on the structure of that particular bacteria. So fun stuff. All right, so here's the back to pylori on stomach cells right here, just showing those guys on this particular slide. Shows the flagellum, those little appendages, it helps it move. Another key. All right, so now we're finished with the proteobacteria, right? So we're finished with the proteobacteria, or should I say, let's do the monodota. So now we're going to talk about the other gram eggs, including the Endomyces, chlamydia, the bacteroides, the cytophaga, the fusobacteria. We're going to also talk about the purple and non-purple, like the purple and green non-sulfur bacteria, cyanobacteria, spirochetes. All right, fun stuff. Let's jump into the first one. You've heard of cyanobacteria before. Maybe. I think so. In my mind, you did. But the reason why I said that is because you live by water. And we talk about blue-green algae, right? You know about algae. You live by water. So cyanobacteria are really cool because these are going to be called the oxygenic photosynthetic bacteria. A lot of words just to basically say they carry out photosynthesis, the regular photosynthesis. So whenever you see, bless you, carry out oxygenic, you're welcome, oxygenic photosynthesis, that's regular photosynthesis that uses oxygen as its final electron. Well, that's going to be taking up oxygen and taking up water, and it's going to be, well, taking up carbon dioxide, taking up water, taking up sunlight to then make your oxygen, your sugar product, your glucose. So when looking at this particular reaction, we're seeing the water being taken up, the carbon dioxide, your light. It's going to make your sugar. So the reason why this carbohydrate equation is simplified like this is because you have for every carbon to one water molecule. That's essentially what it's showing. If I were to put like a 6 behind it, that would be glucose. 6,6H12O6. But it's just saying what if it was like a pentose of some sort, right? So different types of sugar molecules can be made here. And so then you have water and you have oxygen. That's photosynthesis, right? The last time I checked, the only thing that's a little bit different about that is the water that comes out on the other side of that particular reaction, right? That's the only thing that looks a little bit different on this reaction. So when talking about the cyanobacteria that contain heterocysts, which are like these little wall structures, the filamentous walled cyanobacteria, and they can fix nitrogen. What does that mean? Fix nitrogen.
Change it into something useful?
Yes, it can convert atmospheric nitrogen into a useful form, right? So atmospheric nitrogen into nitrite, nitrate, ammonia, all right? Useful forms. And then these guys have gas vesicles that provide buoyancy to allow it to float, to be able to be present in the water bodies. They can be unicellular. They can be colonial. What does that mean? Colonial means that like you can have a circle, a cell of bacteria inside of like in a cluster. So like colonial, like colonies, colonial, that's what that means. And then filamentous means that you're gonna have like those little those appendages, like those little long, like skinny appendages, not appendages, but long, lengthy structures. All right, and then these guys are important because we believe they played an important role in the development of life on Earth by significantly increasing the O2 concentration of the atmosphere because the early life was devoid of oxygen. It had the anaerobic gas, those inorganic gases like the methanes, the sulfur, but not necessarily the oxygen just yet. So these guys, we believe, came along to play a big role in, a big part in making or creating that oxygen. So let's see what they look like. All right, so looking at the cyanobacteria, we have these two figures right here in our text, figure 11-13. And so looking at these guys, we're still talking about gram eggs, right? So if we look at the structure of these guys, we'll see over here on the left-hand side, part A, you have the filamentous cyanobacteria. And so you have the little linkages, the long-like structures I was telling you guys about. You have heterocysts that look a little bit different, right? So these look more like rounded, whereas these look more box-like, right? These look more box-like, like almost plants. They look a little bit like plant cells, but they're not plant cells, right?
Similar to algae?
Yes, that's a great question. That's a great statement. That's because cyanobacteria are very similar to algae because they are blue-green algae, right? So you have blue-green algae, which is what we call cyanobacteria. But you also have like red algae, you got brown algae, green algae when talking about our protists. All right, so they're similarly related to our protists and our plant cells too, because plant cells carry out photosynthesis too, right? So they look a little similar to these guys. Now the heterocysts, you can see them because they have the more rounded edges like that you see. They're pointed out right there. And these guys can actually fix nitrogen and convert it into useful form. And so this is the unicellular form of cyanobacteria. And so this particular one is Profluorococcus, which is the most abundant photosynthetic organism. And so this particular one is unicellular, is non-filamentous, right? It doesn't have those extensions on it like that. And that'll be a type of, or a form of this particular bacteria. So what does this tell us? It shows us that bacteria can exist in multiple forms and still be the same bacteria. Right? The same group of bacteria, is different forms of that bacteria. All right, we're going to switch gears now to talk about the anoxygenic photosynthetic bacteria. What does that mean, anoxygenic? What do we think that means, anoxygenic? Without, right? Without oxygen. So these guys, the phylum Chlorobi and the Chloroflexi, these guys are going to carry out photosynthesis, but without oxygen. So if it doesn't use oxygen, what can it use?
A lot of other stuff.
A lot of other stuff, right? Some inorganic things like sulfur, right? Other things like, for instance, the variations of nitrogen, all right? So let's see what that looks like. So here we have this chemical reaction where they're using H2S. What is that, H2S? It's a sulfur. It's hydrogen sulfide, all right? So H2S is hydrogen sulfide, if you want to write that down. It's hydrogen sulfide plus the CO2, which is our carbon dioxide. So hydrogen sulfide, and what's really interesting about that hydrogen sulfide, it's a lot like water, right? That's why it's an easy kind of shoe-in. That's an easy shoe-in for this particular case, right? Looks a lot like water, it's just not water. And so with the hydrogen sulfide, it interacts with that carbon dioxide and the light energy, and that's going to then yield that carbon molecule that I was talking about, that carbohydrate of some sorts, whether it's glucose, lactose, sucrose, I don't know, different types of oses. And then you have your water, and then you have the SO, elemental sulfur, that's going to be produced. So in this particular case, your final electron acceptor molecule is not going to be oxygen, right? Because there is no oxygen. There's no oxygen, so it's not the final electron acceptor molecule. So there's going to be a sulfur, this particular example. All right? So when looking at this, what are the bacteria? We're going to talk about these on the next slide with a chart for those guys. Green sulfur bacteria. I want you guys to be able to differentiate between green sulfur bacteria, green non-sulfur bacteria, and then your purple sulfur and purple non-sulfur photosynthetic bacteria. All right? So when looking at this example, it's really just these guys right here. Green sulfur bacteria, the phylum Chlorobi, and then your chlorine salt non-sulfur bacteria, Chloroflexi. Let's look at those guys on the next slide with a nice little table for them to differentiate between them. All right, so looking at this slide, we have the selected characteristics for these photosynthetic bacteria. The first one, it shows cyanobacteria. Then you have all the different variations of like green and purple sulfurs. Then you have the examples, the phylum that it's going to be associated with, comments about it. We're going to talk about the electron donor for carbon dioxide reduction and whether or not it's anoxygenic or oxygenic photosynthesis. So for cyanobacteria, an example is Anamina, and this is a cyanobacterium. And the thing with this one is that it carries out photosynthesis like plants do, like I mentioned before, regular old photosynthesis. So that tells us that it's oxygenic, right? This is going to be oxygenic photosynthesis. And we're looking at this one, it's going to use water as its electron donor for carbon dioxide reduction, right? So water is the electron donor, oxygen will be the electron acceptor molecule for that. And then we have the green non-sulfur bacteria. So these guys don't have sulfur, as the name implies. An example is Chloroflexus, and they're in the Chloroflexi phylum. And so they grow chemoheterotrophically in aerobic environments. Okay, cool, because many of our gram negatives do grow chemoheterotrophically, right? Especially our gamma protein bacteria, they're chemoheterotrophic. Cool. And so these guys get their energy source from organic compounds. Okay, cool, just like chemoheterotrophs should. And then these guys are anoxygenic though. All right, they're anoxygenic, so they don't need oxygen for this chemical reaction to take place. All right, and then green sulfur bacteria. Okay, so here we go to differentiate between these guys. How are they different? Okay, well, the difference between these guys is that, well, of course, they're not going to have the sulfur present within them, but this one is going to have the sulfur granules for the green sulfur bacteria, and that's going to be present inside the cells, and that's why hydrogen sulfide would be this electron donor molecule, and this is going to be anoxygenic photosynthesis, right? Because there's no oxygen present. There is no oxygen present. And so now we have our purple bacteria. So we have purple non-sulfur, purple sulfur bacteria. Rhodosporidium is actually really cool. So rhodosporidium grows red. It's pretty cool. And it's in the phylum Proteobacteria. You have Prometheum, which is also in the phylum Proteobacteria. And the differences between them is that one has sulfur, one doesn't, right? One has sulfur, one doesn't. But nonetheless, though, these are going to be anoxygenic photosynthesis because they're not using oxygen as their source for the electron donors. All right? So that's a very simple way to break those guys down. All right, checking out the whether or not they're anoxygenic or oxygenic and their electron donor capabilities. Yes, ma'am.
So for the non-sulfur, what's the final electron except for the various?
Different types. It just depends on what's there in the environment.
Mm-hmm.
It can vary. If it's like a nitrogen type compound, even sometimes carbon, different types of carbon variations too can be present. Just depends. All right, so now to the fun stuff, chlamydia. All right, so chlamydia is really cool because chlamydia is a gram-negative microbe, and it's really interesting because this one grows intracellularly. It doesn't have a peptidoglycan in the cell wall either. So if it doesn't have like a peptidoglycan in its cell wall, that makes it kind of hard for certain antibiotics to work for it, right? Because some antibiotics target the peptidoglycan of that bacterium, right? So it's kind of hard for that to work against that one. So it grows intracellularly. What does that mean? Intracellularly. Inside of a host cell. Need a host cell for this one, right? Is it living? Absolutely. It just needs a host cell. Okay, so when talking about chlamydia, what makes it so cool? Okay, chlamydia is cool because it has different types of like small structures that helps with its infectious abilities. So that's elementary bodies. That's going to be the infectious state. So you have an elementary body. I'll write that on the board. And elementary bodies are referred to as EB. Elementary bodies are referred to as EB. And then you have reticulate bodies. All right, that's two different things. So we have elementary bodies, and then we have the reticulate body. All right, and so we're going to make sure that we're differentiating between the two. And the difference is that the elementary body is infectious. All right, the elementary body is infectious. That's what you're going to be seeing. That's going to be attaching to those, affecting those mucous membranes and causing those infections. And then I'll hang on to the reticulate body in just a second. But I'm going to come back to that one in just a moment because I want to talk about the types of chlamydias. So with the chlamydias, you have chlamydia trachomatis, which is the one that causes the STI because it's going to cause trachoma as well as urethritis. So the urethritis, which is the inflammation of the urethra, which is the opening for the release of urine, right? And so when talking about this particular microbe, right, you have different types of chlamydophylla. So chlamydophylla Potaceae, this particular one is going to cause respiratory participiosis. Always trip up on that particular word. A little bit difficult to say that one. All right, and then you have the chlamydophylla pneumonia. All right, so when talking about that particular one, that one's going to cause some type of pneumonia in young adults. All right, so the thing that I want to get to for these guys is the picture right here. All right, so here we have, I'm going to zoom it over. The bacterium is going to affect the host cell, it's going to attach to that host cell's wall, and ultimately it's going to be phagocytized or taken up through invagination of that particular host cell. It's going to be inside of a vacuole, and once it's inside of the vacuole, then it's going to become a reticulate body. So as a reticulate body, this is the non-infectious form, but it's the replicating form though. So it's non-infectious, but it's replicating. The infections is going to be caused by the elementary body. The reticulate body, non-infectious, replicating. So what does that mean it's doing inside that cell? It's doing what? Dividing. It's dividing, it's making copies of itself, right? So as this particular cell, the reticulate bodies are dividing, this is going to ultimately divide continuously, making lots and lots and lots of reticulate bodies. And so as the division process takes place, you get a lot of them, and some of them will differentiate into elementary bodies. All right, so some of them will differentiate into elementary bodies. They go back to the original state of where you see right here with the little small circular form. That's because those are going to be ultimately released from that cell. And when they're released from that cell after differentiating, they're going to be out to infect more of those whole cells. All right, so the elementary bodies are released to go out and infect more and more host cells. All right, and this cycle continues to where until it's treated. This continues until that particular infection is eliminated, to where it's going to continuously make more and more copies of itself using that host cell's machinery to do so. All right, so this whole process takes about 48 hours to complete. for the chlamydia life cycle, about 48 hours to complete. Alright, so looking at this one, here you see the micrograph of the chlamydium and the chlamydia, and so when looking at that, you have the structure. I'm gonna zoom in over here. So you have the elementary body that's gonna cause the infection itself inside that whole cell, and then the elementary body will ultimately be developing into the reticulate body, so it's gonna divide, it's gonna be making more and more copies. You see all these reticulate bodies that's present in the lighter stage colors right here, so a little darker structures, but the lighter interiors, because you have the intermediate bodies. So the intermediate bodies have the more dark, dense centers in them, and that's like just the intermediate, the middle between reticulate and the reticulate and elementary. That's all, intermediate in the middle. That's it. But nonetheless, though, that's going to be a type of bacterium that's gram-negative. It grows intracellularly, and it's going to cause a very well-known STI. All right, so these are actually pretty cool. The planktomycetes. Okay, so the planktomycetes are so cool because these are budding bacteria. Budding is not really something we see with bacteria, right? We've seen that somewhere else where budding could take place off that particular cell, but it's not that common because budding is typically for fungi. It's asymmetrical cell division. Okay, but when talking about this particular bacterium, DNA analysis places them among bacteria, but their cell walls look like archaea. Their cell walls look like archaea, so it's like, and they have an organelle that resembles the eukaryotic nucleus. So it looks like the nucleus. It doesn't have an organelle, so sorry. It doesn't have the organelle, but it looks like a nucleus. It has a nucleoid region, right? So this particular cell resembles the archaea and it resembles the eukaryotic nucleus because of the nucleoid region, and it has this double membrane-like structure around it to resemble an organelle. So when looking at this particular example, you have Gemata aspergillobus. I've always messed that one up. So Gemata aspergillobus, this particular one has that membrane around the DNA, and it looks like a eukaryotic nucleus. So what does that tell us? It tells us that it kind of supports that idea of endosymbiosis that we mentioned before about taking up those membranes and then having that double membrane bound structure because it was taken up in the early precursors to cell development. So it kind of supports the idea of organelles forming from being taken up by bacteria. Right, so this is a bacterium that kind of goes along with that idea. And so then we have another group of gram-negative bacteria. We have the bacteriotas. And so within this one, we have the bacterioides, which are found in the mouth and the large intestines, with the cytophaga, which are also really, really cool because they break down cellulose. So they break down cellulose. They can break down cell walls, right? And they can break down fungal walls too. They break down chitin. So they break down cellulose and chitin in the soil. But this is the one I wanted to get to, the fusobacteria. So this one actually has a really distinctive smell. This is like they're long, slender, thin bacteria, the fusobacteria. They're anaerobic and they're found in the mouth. So with that particular bacterium, this one can cause dental abscesses. So we know that some bacteria can cause cavities, right? So Streptococcus mutans causes cavities. Cool. This one can cause dental abscesses. So like if your oral health isn't the best and you like have like not improper flossing, caring for your gums, it can cause these different abscesses in your mouth. All right, so this is the bacterium that's going to cause that infection. That's why they give you an antibiotic for that type of thing, right? Because it's a bacterial infection. So we're looking at these guys. Make sure that we are familiar with these bacteria, specifically the fusobacteria. All right, so specifically the fusobacteria. Make sure you guys remember that one. All right, and then we'll talk about the spirochetes. So spirochetes, they should be what type of shape? Spiral. All right, they're on the spirochetes, the tightly wound shape, right, for these guys. And so these are coiled and they move via the axial filaments. And you know, the axial filaments is going to be on the inside of it. It's going to help it with that rotation-like movement. And it's going to then include our treponema pallidum, which is the cause of agent of syphilis. We've seen syphilis on our slides in the lab. All right, we have Borrelia, which is going to be causing relapsing fever and Lyme disease. And then we have leptospira, which is going to be secreted in animal urine. So that particular one is not going to be for us, but it's going to be secreted in the animal urine, and it can be spread to humans though through contaminated water. So leptospira can be spread to humans, unfortunately, through contaminated water. All right. All right. So when we look at the spirochetes, this should look a little familiar from chapter 3 itself. So we talked about this type of figure right here before, where you have the axial film that's going to be attached at one end of the cell. And then the sheath covering is going to be on the outside to protect. You can't really see the filament because it's on the inside. Whereas for a flagellum, you can see the flagellum on the outside of the cell. Right? This one can't see it, it's on the inside. So the sheath covers it, protects it, and it's gonna have that whip-like motion to propel it through medium or wherever it's gonna be at in that environment. And so this cross section right here will show you, if you look at it on the top, you cut it and you look down onto it, you have the sheath on the outside, that's what we're gonna see. And so within that, you have the cell wall that's gonna actually contain the cell components on the inside, so like the little nucleoid region, it's little ribosomes, it's cytoplasm, all that great stuff. And the actual filaments on the outside, that's gonna be helping with the movement attached on that one end. Alrighty? Alright, so, just questions, things for you to think about. I would go through and answer these questions too for you guys, that way you have like a little thing to look at, to practice with. Alright, so now we're gonna switch gears. Alright, so now we're switching gears because everything so far has been gram-negative bacteria. Alright, everything so far has been gram-negative bacteria. So now we're gonna go into the gram-positive bacteria, and we're gonna talk about the genre of the Bacilliota, so that's gonna include Bacillus, of course, the Mycoplasmatodes, the Dinococca, and then we're gonna talk about the actinobacteria as well. Alright, so the first thing we're gonna do though is talk about how we normally divide gram-positive bacteria. So we look at the guanine to cytosine ratios, right, G to C. So the G to C percentages is gonna be very important. So we look at the ratios to differentiate between our bacteria. So whenever there's a high G to C ratio, meaning that it's gonna be greater than 60%, it's gonna be the Streptomyces and the Mycobacterium. So looking at Streptomyces specifically, this means that when you look at it, your G to Cs, right, because G to Cs, 3 hydrogen bonds, all that good stuff. So all the G to C pairings that's gonna be within your DNA. Right? So what's the ratio that we see the G's and C's? Right? What's the ratio for the G's and C's? And so when you have this at 69 to 73% Streptomyces, what do we know about Streptomyces? This one is going to be the one where we get most of our antibiotics from. Right? This is the one we get most of our antibiotics from. 2/3 of the world's population of antimicrobials come from this guy. Right? It's a soil microbe. We're going to talk about it. It's going to come back. Then the Mycobacterium, you've seen that. You've seen that, right? So when looking at the Mycobacterium, this one is going to be 62 to 70% G to C's. Now you have your low G to C ratio. This is going to be less than 60%. So Streptococcus, that's where you're going to find the strep throat. Strep throat's down there. All right, so Streptococcus, G to C constant of 33 to 44%. And then Clostridium, that bad one, right? This is going to have the G to C content of 21 to 54% ranges. All right, so let's jump in. Let's go to the first one with the low G to C gram-positive bacteria. So we're looking at the Clostridiales. Clostridiales is a order, right? ALES is going to be an order. And so it's going to include Clostridium, which we know is what? Going to be an endospore containing bacteria. So what does that tell us about this one? Is it easy to kill?
Yeah.
No.
Oh, wait.
No. I flipped the question. Normally I ask, is it hard to kill? Are you paying attention? All right, so it's not easy to kill, right? Very difficult to kill this one. And so when talking about the endospore producing microbes or obligate anaerobes, what does that mean? They need to grow without oxygen, right? They need to grow without oxygen. So these guys do not like oxygen, they don't want to see, they don't want to be around it. And so what do they include? They can include Clostridium tetani, Clostridium botulinum, and Clostridium perfringens, which we've all talked about before. All right, so when talking about these guys, these also unfortunately can be used as bioterrorism type weapons because of their difficulty in killing, right? And so C. diff, Clostridium difficile, that's the one I've been telling y'all about. I meant to tell y'all, there's actually, somebody asking us, how do you get the money for like the fecal donations? You gotta Google it. I could pull it up. I looked into it one time. I counted myself out because of like the rules. I was like, dang. It's like if you have like gastrointestinal issues, they don't want yours. I'm like, oh, y'all should take mine just to do some more work on it. But nonetheless, right? This one, C. diff, is a very serious infection. It's typically attributed to being overprescription of antibiotics, right? Because C. diff can naturally exist, but if you take antibiotics that kill off the good bacteria, that's going to make the C. diff proliferate, outnumber the good bacteria, and you're going to regret that. You're going to get very sick. That's the hospital stay. That's the hospital stay because they have to give you fecal transplant. Right? That means that they're going to give you, I know someone this happened to like about 2 summers ago. They had to give them a fecal transplant. They actually have a fecal samples, and they have a tube outside the body, and they're going to pass the fecal matter into the gut with the tubing to get their gut flora back up. It smells quite atrocious. So, it's a bad smell. But when talking about the next one though, the Epilopicum, this particular one, you don't need a microscope to look at it. It's so big. You can look at it with your unaided eye. All right. So when looking at this one, what makes it unique though, besides the fact that it's so big in comparison to other bacteria, is that its starter cells form within the parent cell. Within it.
Almost like a colony?
Almost like a colonial bacterium, right? So they form within the bacterial cell. So it does not do binary fission, because binary fission is one cell split to form two cells, right? So this is going to divide on the inside of that cell. So it shows you the size comparison. Paramecium, you guys remember paramecium, right? Paramecium is like kind of small. They're fast too, right? Paramecium. But these guys are fairly small in comparison to the Echinopithecum. That's big bacterium, right? That's a nice size bacterium. All right, so the next order that we're looking at, because notice this particular category, the Bacilliota, includes the Clostridiales as well as the Bacilliales. So these are going to be kind of similar but different. So that's why we were talking about how these guys have endospores together. Bacillus and Clostridium have endospores. So they're both gram-positive. They're in the same, not the same order, class. So they're going to be in the same class. Why they're going to be closely related. All right. So bacillus, we know a lot about bacillus. It's going to be an endospore producing bacterium, it's a rod-shaped bacterium. These are common soil inhabitants. It's normally found in the soil. Bacillus subtilis. You have Bacillus anthracis, which is the causative agent of anthrax. All right, the causative agent of anthrax, which is a bioterrorism weapon, right? And then we have Bacillus thuringiensis, which is pretty cool because Bacillus thuringiensis is an insect pathogen, and people use it. Some farmers use it. It's called a BT gene. So if you look it up, you want to look more into it. There's a gene called BT that you can actually put in your plants. And if it's in your plants and the bacteria and the insects try to eat those plants, they're going to pick up that gene, and that gene's going to make them, that particular pathogen is going to make them very sick. All right, Bacillus thuringiensis will make them very, very sick, kill the insects, and it's kind of like an insect control mechanism. So that's going to be used for agricultural purposes. And then you have Bacillus cereus. Okay, so this is actually really interesting because it do cause food poisoning. Do you guys like to eat rice?
Yes.
Same. I love rice, right? Now, do you eat rice after letting it sit out?
Oh.
Why do you not want to do that? Because it causes that. Yeah, don't do that because with your rice, okay? With rice, once you eat it, and if you don't eat it within a timely manner, like let's say it drops back to like room temperature, if you eat it, it's hot, it drops back to room temperature, put that in the fridge right away. Don't let that sit out for too long because that will allow for the bacteria to start back growing, right? Because remember, the refrigerator is heating food should kill the bacteria, but that's not always the case because remember, it's bacillus, right? Bacillus, can you eat bacillus and kill it on the stove? You can. It's really funny. So with bacillus, you have to kind of steam it and you have to heat it, and that's an autoclave. You don't have autoclave at your house. You don't have an autoclave. If you do, let me know because like I could come over and do some stuff there instead of having to come to work. So with that being said, bacillus has the endospore present. And when you cook it, it's gonna make it to where it's not gonna be an active form of that particular bacterium. So that's why when you let it sit out for a little bit, make it drop down to room temperature, the bacteria can start back growing because microbes grow at room temp, right? And so the microbe can start back growing, and if you consume that, once it overgrows with Bacillus cereus, you will be very sick. You will not be happy. You'll have food poisoning. All right, so that's why that is misinformation. So I was gonna tell you guys about. There's an article floating around right now. It's misinformation. It's talking about how this person had ate some rice that was that stayed out too long and they lost their legs. It was a whole lot of stuff with that. But they really did lose their legs. They really did lose their legs from cellulitis, but like that was a whole other thing that goes along with it. It wasn't just from this. All right. So Staphylococcus. We know Staphylococcus aureus, Staphylococcus epidermidis, right? So this one is also in this order. So it's like, oh, okay, in the same order, but they're still different because bacillus are rod-shaped, Staphylococcus are coccyx-shaped, right? And so these guys form the grape-like cluster because the staphylo, the grapes, the coccyx, a circular structure. So we know about Staphylococcus aureus. It's a bad one, right? It causes wound infections. It's often antibiotic resistant, so it's resistant to penicillin, methicillin, vancomycin, lots of different types of resistance to antimicrobials. And it also produces an enterotoxin. So this particular one can produce an enterotoxin, and that could lead to, that's why you have to make sure you kill the bacterium and kill the toxin, because that one can lead to a lot of issues with cellulitis, swelling. With this one, you could go into toxic shock syndrome. So many different things that could happen for exposure or infection with this one. All right. So with Staphylococcus, that's a picture of it. Oh yes, that's a picture of Staphylococcus aureus right there. The yellow cocci-shaped bacteria, and then we have the endospore present. The endospore with a little spore coat makes it very difficult to kill that, and also makes it hard to stain it too, right? That's why we did the heat for the malachite green for the endospore stain. It took a little while to do that. You have to apply the heat to that particular microbe to make sure that you get it stained properly. All right. And then now we're looking at still in this same class, we're looking at this particular order here, the Lactobacillus. So this one's going to be aerotolerant anaerobes. What does that mean? They tolerate oxygen. They prefer to grow in the absence of oxygen. They're not going to use it though. They're not going to use it because they prefer to grow in the absence of oxygen, but they'll tolerate it. And so these guys make lactic acid from simple carbohydrates. So yeah, that's a pretty cool process. Lactic acid is going to be fermentation going on right there, then, right? And so with this particular order, it includes lactobacillus. We know lactobacillus can colonize the body, different parts of the body. This one can be found in the bathroom area because it's going to be a natural one that just hangs out down there unless there's going to be an offset of the pH. And so they're also used commercially in food production, right? Milk, yogurt, yep. All right, so here's a fun one, Streptococcus. It's not that fun, but it's cool to talk about it. So when talking about Streptococcus, this particular one falls within the Lactobacillus order. And this particular microbe, we know it's spherical in its structure. We know that already, right? We know its shape. We know that this one is going to be arranged in the chains, the strepto portion of it. And so they're going to be making enzymes that damage tissues. Right, so Streptococcus produce different enzymes that damage tissues, and we're able to see what this looks like when we use blood agar. So blood agar is a type of complex medium that we use to differentiate different streptococcal strains, right? So when talking about blood agar, it actually includes red blood cells from sheep. So it's like TSA with 5% sheep's blood, all right? And that's why it's red in appearance. So when talking about the bacteria, you have the alpha hemolytic bacteria, you have beta hemolytic bacteria, and then you have gamma hemolytic bacteria. So beta hemolytic means that these guys are going to completely hemolyze or destroy those red blood cells. They're going to completely break down those red blood cells. So beta hemolytic breaks down the red blood cells, and we can differentiate those because your Streptococcus pyogenes and Streptococcus agalactae are going to be the ones that do that. So Streptococcus pyogenes, pyo pus formation, that's going to be the pus that you see on your throat, your Streptococcus pyogenes, strep throat. So it can cause pharyngitis, impetigo, scarlet fever. You can, if you don't treat it, you can unfortunately you can have scarlet fever is the one that gets really bad, right? So you have to make sure you treat strep throat if you do have it. So Streptococcus agalactae, neonatal sepsis. So we know that with sepsis, it's going to be the presence of the microbe within the blood. We don't want that, right? And also meningitis. So bacterial meningitis, we know is the meningitis present in the spinal fluid. So we have to eliminate that as well. You don't want anything in your spinal fluid but things that's supposed to be there. The normal cells are supposed to be there. Nothing bacteria should be there. So alpha hemolytic means that these guys partially break down those red blood cells, right? So beta is complete hemolysis. Alpha hemolytic is going to be partial hemolysis, not always breaking down those red blood cells. And so that's Streptococcus pneumoniae, which is going to cause pneumonia. That's a BSL-2. You can work with that one. Streptococcus mutans. You can work with that one too as a BSL-1 sometimes. It can also be a BSL-2 depending on where you're getting it from, what you're working with it. So Streptococcus mutans is the one that causes dental cavities, holes in your teeth. All right? So that's why it's so important to brush and floss, get the food out of there because think about it. Let's say, for instance, you go home, you eat your dinner, and you don't brush your teeth before you go to bed. What happens is once you go to sleep, that's perfect. Bacteria is so happy. They're like, ooh, all that food, all those resources, the nutrients, I'm happy. They're going to divide. And when they divide, they're going to be on your teeth, right? And so if you have any type of, not so much as deformities, but like weaknesses within your teeth from you not taking care of your teeth properly, that'll lead to those holes start to appear. Now, normally, where do holes normally appear in your teeth? In between them, right? You don't normally see them on the front. That can happen. You can have them on the front of your teeth, but they're normally in between the teeth because that's where you normally don't floss to get the food out of there. And that's where these bacteria are going to be hanging out and getting ready for the party. All right? So, Enterococcus. So, we've seen Enterococcus. This is going to be found in the intestinal tract, and these are going to be a nosocomial type infection because these guys can infect you after having surgery, right? Surgical wounds, and the urinary tract too. So, dang, the catheters, right? These can be found on those catheters. So you have Enterococcus faecium and Enterococcus faecalis. These are some interesting microbes you have to pay attention to in hospital settings. You guys heard of listeria before, right? Yeah, you see in the news a listeria outbreak from fruit normally. So listeria monocytogenes contaminates different foods. So it actually contaminate like cantaloupe, mangoes, melons, fruits. I like mangoes too, but like it's just that it has to be present. It's not gonna just like be hanging out with the fruits. It has to be like contamination, spread, cross contamination some type of way. All right. My mom actually had listeria. She was at a hotel in Florida, and on the salad, on the fruit bar, they had the contamination from that, and she got sick off that fruit bar. So yeah, it was almost that was cool. That was kind of cool. She gets so mad when I say that, but she gets so mad. But the next one is gonna be mycoplasma. So when talking about mycoplasma, this particular microbe is really interesting because it's going to have it's in a different order. So with this one's the Mycoplasmatoda, and it's gonna have a cell wall not there. So it's gonna lack a cell wall. So because they lack a cell wall, we call it pleomorphic because it can take on many type of shapes, right? It can vary in its shape and its appearance because it doesn't have a cell wall. The cell wall makes it rigid, right? Makes it rigid. So these are probably the smallest self-replicating organisms that are capable of free living existence. Research needs to be done on that more, right? It's not just we know this 100%, but it probably is. The reason why I say probably is because we haven't even explored the ocean fully. We still have so much work left to do. There's so much work, so things to think about. So Mycoplasma pneumoniae causes mild pneumonia. So you probably think when looking at this one, it's going to be related to some fungi, right? In this particular way that we talk about it, it's like not necessarily for it's just the similarities in the way that we describe that. All right. Another thing I want to add is that the reason for that is because it typically when you grow it on artificial media that have sterols in them. All right. You grow this on artificial media with sterols because these guys actually will have sterols present, and so sterols aren't really found in the cell walls of bacteria. Sterols are found where? You guys, it's way back in chapter 3. Way back. No, sterols. I love that. She was like, yeah, absolutely not.
Blood?
Not blood. It's something that we haven't. We're gonna hit this in later chapters.
Plasma membrane?
It's gonna be the plasma membrane of something. Typically,
Lungs?
huh?
Lungs?
Not lungs. What'd you say? Fungi.
Wow.
Yeah. So you typically grow it with artificial media that contains the sterols in it. All right. So you have the artificial media with the sterols in it. All right. So we are done with the low G plus C bacteria. Now we're going to switch gears and hit the high. I won't say A. The reason why I'm saying A is because of the actinobacteria. So the actinomycetoda, this particular group this particular clustering of the microbes are going to be high G plus C, so the ratio of 60% or higher, right? These are still gram-positive. They're highly pleomorphic, so their shapes can be variable with these guys. And some have branching filaments. So remember how we saw like the little individual cells kind of like lining up to look like little extensions or appendages almost? And many of them are found in the soil, like a lot of our bacteria, as you guys can see, right? They're soil inhabitants for the most part for this particular group of microbes. So the first one, Mycobacterium. Okay, so we know that one. We've seen that one. Mycobacterium tuberculosis. Yes, sir. Yes, ma'am. That's going to be a problematic microbe. So Mycobacterium has a cell wall with a very thick acetylglycan layer, and that's why you can mess around and gram stain is positive. It's not supposed to be gram stained because it has a waxy mycolic acid in its cell wall structure, so it can give you that false positive-like thing. But we don't really gram stain it. We want to do an acid-fast stain instead. All right, we want to do an acid-fast stain instead because you want to be able to show definitively, like, oh yeah, this is Mycobacterium tuberculosis, or this is Mycobacterium, what's another one? Smegmatis. So when looking at this one, it grows really slow. That's why whenever they ask you, like, have you traveled outside the country in the last 30 days? That's because this particular one takes a little while to incubate and to grow. I used to work with Mycobacterium smegmatis in the lab, and that one it would take me a whole week just to get it to grow. All right, so if it takes a week just to get it to grow on media, then it can take a little bit longer to grow inside of your body. So about 2 weeks, depending on the person. Oh, I just realized I'm not recording. Dang, that means I gotta record this. Okay, so it just dawned on me. So we're talking about Mycobacterium tuberculosis that causes tuberculosis or TB, and then Mycobacterium leprae causes leprosy. Remember I was telling you guys about how I, didn't know out here, like, you have to take a TB test to even work out here. That's not a thing in Louisiana. I was just out there working. Minnesota, working. Texas, just working. No TB test. My first time doing a TB test since I was in, college. Hadn't done it in a long time. Y'all like, dang, y'all, it's a jungle out there. But when talking about leprosy, the thing I was gonna throw out there is that Louisiana, in Carville, Louisiana, we have the only leprosy sanctuary, and it's actually still there. So if you ever want to look into that, Carville, Louisiana, Carville, Ville, it's the only leprosy sanctuary. They'd actually like shun people to that area, so that way, because they had leprosy and like they wasn't really sure how it was transmitted back then. It's just that it caused a lot of sores and lesions on the skin, so it looks kind of bad to the eyesight. So they really would shun people out to that area. So that's the only facility in the state, in the U.S. actually, where you can go or you could go. I don't know if it's still active. I know it's still there. I know it's still there. I don't know if they're still taking people, but I know it's still there. Because leprosy is not a thing, right? Here in the U.S. of A. now, not that common of a thing now. All right, so the next bacterium, Nocardia. So Nocardia is pretty cool. So Mycobacterium is acid-fast positive. Nocardia is also acid-fast positive. All right, so Nocardia is also an acid-fast positive bacterium. It forms fragmenting filaments, and this particular one, Nocardia asteroides, causes pulmonary infections, so upper respiratory infections. We also have Corneobacterium, so that's why it's so important to write out stuff, right? Corneobacterium diphtheriae, that could be confusing with Clostridium sometimes when you look at that particular write-out for that. So this one. And so then you have Propionibacterium. I told you guys about that one before, because this one can form propionic acid, and propionic acid can actually be used in a lot of, what's that stuff? Cosmetic products. But also, Propionibacterium can actually cause acne. So Propionibacterium acnes is the cause of agent of acne, which is why you can take clindamycin to clear that up, if you're not allergic to clindamycin. Yeah, unfortunately, so that means that the gram staining isn't really ideal for this particular one because it could give you some false results on that one. So additional testing is needed, right, to talk about what that one could be. And then by being highly pleomorphic plays a role into that, the different forms and shapes in which it can appear can possibly be another way to misidentify this one.
So is that changing the thickness of the peptidoglycan?
No, not so much. It's just more so with the, you know what? That's a good question. I'm answering that thinking that it shouldn't change the peptidoglycan. That should be the, it should be stable. But what if though across that type is pleomorphic? If it's changing in its way that's appearing, then maybe it's shrinking its, I wanna write that down. So I just thought about something. Let's see, does anybody, I'll write on this. I left my part because of the rain, but you know. So, part umbrella, change, and to the pocket. I'm gonna look it up and see. That's a good question. All right, good deal. Thank you. You guys need a stretch break? Any stretch? Is that moving? Any stretch? Walk a little bit? Any stretch break? Are you good? Would you like a stretch break?
Yes.
Yeah. Yes. All right, there we go. Let's stretch for a minute. I'm like, stretch break. Like, oh, you know what? Since we have time, y'all stretch. I'll do this. So it's just more so once it's able to penetrate that, it gets to the period. I can look and see like why.
I'll pair for red one. Live.
Or could we recommend?
I'm more like a nerd.
To wear
Nerd? Nope. No, I'm like a, I'll, you know, yeah, like, I want my like chocolate.
So different.
Chocolate, right,
You think we're
right. Turn it black.
Baby Ruth? Oh.
Bacon, my Germany.
We said
I do.
for the,
You try to hate?
yeah.
Yeah.
There you go.
Yeah, back to the well.
Yeah, I
I
want
wanna
to
see
see
my
that.
sex in
Bacterium.
Yeah.
Baby Ruth.
Oh my God.
Probably the candy. Yeah, it's a silver wrapping. Bacon
Yeah.
eater. What else about you? No, I'm all the fat ones. No, give me all the sugar.
You can't say all the sugar that 50.
That's, is it not?
baseball player?
Really, a lot of sizzling, the nutrients that are there.
Okay.
Yeah, that's a good point. Especially because it's not able to,
Yeah.
That's
destroy
good.
by the
I
response.
watched the
Because
car.
it's inside of the.
Yeah, really?
You don't have other things.
I just started like to down there. I just like the hard.
Yeah.
You know what? And then we sign me up.
Alright, so.
Like our normal.
Okay, here we go.
Laughing, you bent over, you bent over. I love her dance.
This is gonna be silly, that makes sense. So for ground edge, you have the cords, it was still the cords, I knew that part.
That's what
But trying to see why doesn't it go further into the cell?
It has
It's been years and years.
In high school, I was like, Yeah.
Okay,
It
so
was
it's
dense.
it's actually similar to the bacteria phase, but not really, the bacteria phase.
I saw a thing in this girl.
They,
she was like, don't do that. She was in the hospital.
Okay.
Well, again, it's like, how does it know? How does it make it sense to electrical
Edit it up,
polarities of
get it
two
down.
membranes? Is that
I
not
literally
different
can't
than
tell her.
charge? You feel all the way inside?
They're fat burritos.
If you get like a regular.
Just like, let me show you.
This is still going.
Oh, hello.
Well, I should give that recording.
I think we have to read about that.
Shalene.
Like for the profile.
Yeah.
Like,
That's it.
I'll email it to you right quick.
Oh, okay. Thank you.
No problem.
Yeah, it's
Alright, we're gonna come back. Let's see, anybody email me?
Yeah.
I saw your words.
Oh, Alex emailed me.
Bruh, those thick son of a gun glasses. She's like, let me do this every day.
Okay,
how
so,
they're cute.
you like one more, I give it a couple more seconds, it's 11:05.
Oh,
Let's see how bad his vision is.
Oh, so for those of you that are in the lab this afternoon, we're gonna do our lab report, we're gonna have that due on the 24th, so that way I'm trying to see about that. So I'll see about the dates, because, I'm trying to get it to where I can get you guys to bring it when you come back for Thanksgiving. Because I know some people aren't doing Thanksgiving, like they built it into the syllabus, they're not coming on campus at all, so I'm like, I don't know, we gotta see.
So we might
Yeah,
not have
how
school
Wednesday,
Wednesday,
so
so
you're
No, I didn't say that. No, I didn't say that. No, I didn't say that. Some people, I said some people, not me, I'm not those people.
Oh,
I would,
not us.
Y'all say safe.
I would like to keep my gas, you know,
Right?
right?
No,
But at the same time, though, for me, your learning is a priority, so I'm trying to figure out the way that I'm gonna do it. Because we're gonna meet at least one day that week, at least one day. So we'll at least meet one day, so we should have lecture lab. The reason why we need to at least meet one day is because you got the lab practical next week.
Yeah.
That very next week, that Monday, so once you come back that Monday, it's lab practical. And I wanna make sure that you guys have the time to look at the stuff, because that's really the practical, gonna be looking at that media and interpreting that media. So we don't meet both days, we're gonna meet at least once. And I'll have an offensive answer for you guys. The 27th.
Oh, wait, hold on.
Yeah.
Always that Thursday. Always that Thursday. Always a different day though, right? Alright guys, so we're back. Let's get all back on track. So, we convert oxygen from the atmosphere to useful form. Streptomyces. Remember I told you guys. Okay, listen up guys. So streptomyces is isolated from the soil, and this produces many antibiotics. So streptomyces is so cool. When I say many, about 2/3 of our naturally developed antibiotics. And so when talking about this particular process of antibiotics in the soil, this has funded a lot of research that goes on today. There's this group of people called like tiny phages. It's a group, Tiny Earth. Sorry, it's called Tiny Earth. They basically go out and collect soil samples to see if we can discover more antibiotics. And they just track it and they tell us where these different antibiotic species or these different bacterial species are, so we can learn different ways to treat these different diseases that we have. Because remember, I don't know if you guys are paying attention to this, but a lot of the antibiotics that we have today, some of them are becoming quite resistant. Some of the microbes are becoming quite resistant to the antibiotics because we use them a lot, right? So there's why we have first order antibiotics, second order antibiotics, third order, right? Because we have to synthesize them from our naturally created antimicrobials. So we have like semi-synthetic antibiotics because they came from a natural source, right? So that's why we're out doing research to find more antibiotics that could help with treating these different infections. So we have the actinomyces, which form filaments called hyphae. So it's like, okay, that sounds a lot like fungi, right? So these guys have a similarity in their composition to fungi. They're found in the mouth and throat. Streptomyces also has like a little similar composition to fungi as well. So it makes sense too, considering the fact that our first one of our first mass-produced antibiotics was from a fungal source, right? Penicillin, right? So it makes a lot of sense. So let's look at streptomyces. Remember I was just saying that streptomyces looks a little bit kind of like a fungal structure in its nature. So here we have the streptomyces structure. It's filamentous, and it branches off with the hyphae. So we're going to talk about hyphae. Hyphae are going to just basically be these long extensions that exist, and these are going to have these different conidia spores and coils on the surface. So the conidia spore is going to be responsible for making more of that bacterial cell. All right, so whenever you see spores are going to be involved in making something. All right, what it's going to be involved in making, it varies depending on what we're talking about. But this one, we're making more of that particular structure. All right, at the tips of that one. So then you have this particular slide here that shows the cells of the actinomyces that have the little bulbous ends, little dark purple structures that we see there. So like little small ends them. And then we just see an actinomyces over here that shows a cluster of these bacterial cells together. All right. All right. So now let's talk about something that's a little fun to me. So the dinococci, these guys are pretty cool. So the Deinococcus radiodurans, that's like, what in the world? Something sounds like radiation, right? So these guys are pretty cool because they are more resistant to radiation than endospores are. All right, they're more resistant to radiation than endospores are. So it's really, really difficult to kill these guys. And so when looking at them though, this is what's really cool. I would highlight this part. They have a unique arrangement of DNA that facilitates rapid repair of radiation damage. That's crazy because radiation, we know, can actually affect your DNA by causing thymine dimers, right? So radiation, UV exposure can cause the formation of thymine dimers, and these bacteria can actually repair that process, right?
So could these be like really old bacteria from like before our atmosphere was big enough?
Essentially, they could be. We can actually see how old are these guys real quick. So I'll look that up as I tell you about the Thermus aquaticus. So Thermus aquaticus, what does that sound like? Sounds like hot water. Yeah, sounds like hot water. So when talking about these guys, I'm trying to write and type at the same time. I think writing and talking is not good because I wrote down what I said. All right, so when looking at these guys, radiodurans, how old are they? So these guys are, they just found it. They just discovered them in 1956, but doesn't mean they haven't been around. They're much older. They're a lot older. A lot older. All right, so we'll look into that though. I could look into that. They just found them in 1950s, but I'm pretty sure they're a lot older. All right, so Thermus aquaticus is actually pretty cool because Thermus aquaticus was found in Yellowstone National Park in the geysers, the hot springs, right? The really, really hot springs. And so Thermus aquaticus, if you take just the T port of it, take the thermos off, you take the T, you take the AQ, the TAC, right? So that'll be TAC, and this is called TAC polymerase. So remember the process for polymerase chain reaction, PCR? So remember for the PCR process, you had like you heat the DNA, you separate the strands of DNA, And then you have the polymerase, tag polymerase being added. The reason why you add the tag polymerase after you heat the DNA is because these guys can actually withstand really, really high temperatures. So these are thermal-tolerant enzymes. They can withstand really, really high temperatures. And so that's why you add them once the sample heats up. Because then they'll be able to add the polymerase, add the nucleotides to the sequences to extend them out, to elongate those sequences.
But
Alright.
it's not a thermophile, extreme thermophile, it's
No,
just tolerant.
It is a thermal-tolerant enzyme present within it. Yep. So the tag polymerase is very important in PCR reactions, right? When we do it in the labs. And I work with it like so many different times, it's like not even funny. But yeah, so this is a pretty cool thing to know that the bacterium Thermus aquaticus has helped with research purposes, right? Lots of research. So it's pretty cool stuff. All right. And so let's talk a little bit about archaea. They're cool. They're all right. Womp womp. All right. So we're talking about the archaea. What's the goal? The goal is to name a habitat for the different archaea. All right. So just like a moment ago, Regina was like, "Are they thermophiles?" So now we're going to talk about some thermophiles, right? So when talking about the archaea, these guys lack peptidoglycans within their cell wall because they have pseudopeptidoglycan within their cell walls. And sometimes you can see them referred to as pseudomurin. Yep. I was gonna write with my lip gloss. Oh, nope, cannot do that. So pseudomurin does not have peptidoglycan in the cell wall. So it does not have peptidoglycan in the cell wall. Instead, it has the pseudomurin. All right. Now, the one thing about them that's pretty cool is that these guys are often referred to as extremophiles. So this means that they live in extreme environments, right? And so what are some examples of extreme environments? Halophiles. So halophiles require a salt concentration. So halo means salt. Philes means to love or have an affinity for something. They like salt. They love salt. So there needs to be at least 25% of the salt concentration within that given medium for the halophiles to thrive. So the Great Salt Lake in Utah. Have you ever been to Salt Lake City? They have the lake out there. It's so pretty. I was fascinated. I was just driving. You're not supposed to drive and record, but I just had my phone like this and I was just driving. I was like, and I really did. Like, I really wish I had a picture of it. Like, I was just driving like this, trying to watch the road up.
Ooh, your man should have been driving.
I was with myself. Yeah, I know, right? I
was like, hello, you're driving him?
No, I was by myself. But we were over there like, hey, you take the wheel. Let me do this. Let me do my nature thing.
Yes.
Because my family talk about that. They're like, why are you always taking pictures and videos of nature? Let me be great. Okay, when we were in Canada, I was driving and I'm like, they're like, whoa. I was like, well, you drive. Right. So thermophiles, these require a really high temperature, right, to grow. So thermophiles about 80 degrees Celsius or higher. So that's quite hot, right? So they're gonna have to grow in extremely high environments. And we don't work with one here, but I work with one that's considered like a it was considered a thermophile on the low end of the spectrum. And it grows really, really nice. It grows really, really well. It's called Geobacillus thermophilus. So Geobacillus thermophilus, it grows really, really well, but it's also thermophile. So acidophiles. Acido sounds like acids. Yeah. So these are bacteria that grow in acidic environments. So values approaching like 0. That's low. Because our stomach acid is about 2. Right? Stomach acid is about 2. Hydrochloric acid is about like 1 to 2. Battery acid, right? Battery acid is pretty low too, about 2. Don't ever open your battery. Don't do that. Please don't do that. Methanogens. These guys are pretty cool because they make methane, right? We mentioned those before. Remember I mentioned about the landfills. Go to a landfill, you could potentially see, or not see, they'll have some methane producing microbes there and they'll be making methane. You can smell it though. You smell it. These are anaerobic, so they're gonna grow in the absence of oxygen. And you can actually have methanogens present in the gut flora. So some parts of the human microbiota, so like the colon, the vagina, the mouth, they don't have that distinctive smell to it as a result. So remember I was telling you guys about sometimes you can smell certain things on people like infections, or you can smell that particular methane if you have a knack for smelling it. Right? So this picture.
So which one is pseudomurin?
So pseudomurin is gonna be the cell wall. So it doesn't have that peptidoglycan, so that's the pseudomurin instead of that.
Cell wall.
Yes. Yep. Cell wall for your archaea. Yep. don't know if that's,
Where?
I don't know that's.
Oh,
dang.
I said it out, sorry. You could like, I don't know.
No, that was for you, it's okay.
Yeah, I think it's for me, I don't know, alright.
Thank
Alright guys, so with that being said, looking at this picture of the diverse environment for the archaea, just show this figure 11-27, some archaea. Let's move to talk about microbial diversity. Alright, so that we're gonna be illustrating a range of. Alright, so I did this one already with Theo margarita. So Theo margarita nambiensis is the one that I say is like one of the largest known bacteria today. We also have Theo margarita magnifica, the length of like 1 centimeter. I can see a centimeter, you can too. It's small, but you can see 1 centimeter. Alright, you can see 1 centimeter. You have nanobacteria that are so small, 0.02 to 0.08 micrometers. Cannot see that without a microscope, right? So that's a very big difference, right? There's a nice range of bacteria sizes, right? So the bacterial size can vary depending on what we're looking at. Genome sizes too can vary. So we're looking at the genome size for Carcinella rudi, only 182 genes. That's insane. That's not that much, right? Well, especially if you want to think about, for instance, let's see what E. coli's number of genes would be real quick. So the number of genes that's found in E. coli, I should be asking serious stuff like this. Like Siri, hey, no, don't do it. All right, so E. coli has approximately 4,000 to 5,000 genes. So that gives you an idea of that, right? Of how variable that's going to be. So when talking about the Human Genome Project, I told you guys before that they just finished that in like 2003, and we have about 20,000 protein encoding genes, but we have over 40,000 identified genes. So what does that tell you? 40,000 are identified, and only 20-something thousand are going to be protein encoded. The rest of them are not. We don't know what they do yet. We don't know if they make proteins just yet. We have the protein project going on right now. The proteome project is actually going on right now to understand that a little bit better. Right, we have a lot of genes, but like all the genes are not functional as proteins.
Is it like 1 protein to 1 gene?
No, that's the thing about it. So, yeah, so genes can be spliced. They can be cut in different ways. So that gene can be cut in different ways to where it can render different types of proteins. That's the catch. That's the catch for that. So that's why I'm like, there's more work to be done because we don't really know just yet. We don't know just yet. All right, so there's at least 30,000 bacterial species described. That's what we have known to date. But like I said, that number is going to keep changing, right? Because we're still learning things. We still have to explore different spaces. Like the Mariana Trench is the deepest part of the, did y'all know that? The Mariana Trench right now is the deepest part of the earth. That's over there by, is it by Thailand? It's over there by Thailand. And it's quite deep. I forgot how many feet it was, or how many meters it was, but it's one of the deepest parts of the earth, and we still haven't gone to the bottom of it yet. All right? PCR, polymerase chain reaction, we're going to be amplifying segments of DNA. It indicates that perhaps 10,000 or more bacterial species exist per gram of soil. Remember I was telling you guys about that Tiny Earth project about getting out there, getting some soil samples to figure out what's out there? There's a lot of bacteria out there, right? There's a lot of microbial growth going on that we haven't really isolated yet. And so, what limits our abilities to be able to identify these different species? Well, you can't grow everything in cultures. That's kind of hard. So everything is not culturable. Many things are culturable, but everything is not culturable. Another thing is that some bacteria are part of complex food webs and can grow only in the presence of other microbes, right? That provide them with the growth required.